Hi, everybody. Today we are going to talk about proteins, and that is our third and final group of biomolecules. We talked about carbohydrates and lipids so far, and now we've got our last group, which is proteins. So, proteins, as you can see here, are not generally used as an energy source. So, what you should know about proteins and their functions, and that proteins are more about building things. So we might build muscles out of them. They might carry out chemical reactions. We haven't talked about enzymes yet, but these very important molecules in our body, in our cells, called enzymes, are going to be made of proteins. Proteins are part of our immune system. They help fight disease. And proteins are going to be really important when it comes to transporting materials into or out of a cell. So proteins are not, however, a major energy source. Your body generally, under most circumstances, does not break down proteins for energy. Okay, so which of the following is a function of a protein? How well do you understand what we just went over? So choose all that apply. Which is a function of a protein? All right, so let's see how you did. Energy, we just said nope, not energy. Enzymes, yes, that would get a check. Muscles, Plant cell walls, those are made of cellulose, that's a carbohydrate. And cell transport was a yes. So cell transport, muscles, and enzymes. So proteins are different from the other biomolecules we've talked about because they contain carbon, hydrogen, and oxygen, just like carbs and just like lipids. However, they contain something extra. They're the first biomolecule that contains a nitrogen. So that's one main difference. Something else to know is the monomer and the polymer. The monomer is called an amino acid, and we'll take a look at that in a moment. Polymers are called either polypeptides or proteins. Proteins is just the general term for a polymer of these different amino acids. There are many, many different types of proteins, and we're going to be talking about all of them throughout the year. Okay, so another question for you. Using a formula, how can you tell if something is a protein? Take 30 seconds and answer this question now. All right, so how can you tell if something is a protein? If something is a protein, then it will contain carbon, hydrogen, oxygen, just like every other biomolecule, but the difference here is that nitrogen. Nitrogen is something that proteins have that other things do not have. All right, let's continue on. Other examples of uses for proteins include hair, your hair, this dog's hair. It's made of proteins called keratin. Horns, this is made of a protein, so it's not actually bone, but it's protein. Spider silk is a protein. Let's take a closer look at the monomers of proteins. It's called an amino acid. And amino acids have pretty much the same structure, every single different one of them. All amino acids have a group that's on the right of this picture called a carboxyl group. So there's a central carbon, and coming off of that central carbon is a carboxyl group. On the opposite end from the carboxyl group is the amino group. You can remember these different names because carboxyl will have the carbon and the oxygens, and amino, think about the N in amino, has a nitrogen connected to hydrogens. The bottom end here is just a hydrogen, so that's really nothing. And the top end is called a variable group. There are 20 different variable groups that could be present. So 20 different types of R groups or functional groups that are totally different. So every amino acid has the carboxyl group, every single amino acid has the amino group, but every amino acid, there's 20 of them that might be different, and they have one of the 20 different types of functional groups that would go right up here on top. Let's take a look at some of those now. But first, actually, let's have a question. Take some time and draw an amino acid. Okay, so let's see how you did with drawing that amino acid. So the first thing, you've got to draw a central carbon. On one side, you have a carboxyl group. I'm not really too concerned with how well you know the exact chemical structure, but you should know the carboxyl group has a carbon and some oxygens. Over here we have the amino group. So carboxyl, amino. Down here is the hydrogen. And up here, we're just going to abbreviate it with an R. That is the functional group. So the functional group can be one of many different things. So what are some of those functional groups? Well, here we have several of the different amino acids. We have all 20 here. So if you look at all these amino acids, they all look almost the same. They all have a central carbon. Look at that. They all have a central carbon. Doesn't matter where you look, there's always that central carbon. They all have an amino group with that nitrogen. See how they all have that group? They all have a carboxyl group. Carboxyl, carboxyl, carboxyl. Every single one has a carboxyl group. The one difference between all of these amino acids is the functional group or the R group. And notice that in the case of glycine, the functional group is very small. In the case of arginine, right there, the functional group is very large. It might look kind of funny, like in tyrosine. Notice how it has that ring there. Tryptophan, take a look at that. That's a pretty weird looking one. The functional group is different, and each of these functional groups has different properties. Some are nonpolar, and some are polar. Some have a positive charge, some have a negative charge. So they all have different properties that make them unique. All right. Okay, so how do we take one monomer and react it with another monomer to make a polymer? Well, what has to happen is the carboxyl group of one amino acid needs to react with the amino group of another. This is a dehydration synthesis reaction, and the bond that forms is called a peptide bond. So let's take a look at how this might work. So let's take a look at our very first amino acid. In our first amino acid, we've got a carboxyl group that I am putting a box around right now. And our second amino acid, which is over here, here's the amino group that I'm putting a box around. So basically what's going to happen is from the carboxyl group, the oxygen and the hydrogen are going to go away. And this hydrogen is going to go away. And what's that going to form? That should form, two H's and an O, should form water. And so we'll draw in that bond between the N and the C. That is going to be the bond that is formed by dehydration synthesis. That is called a peptide bond. There we see a peptide bond. And now we have one amino acid that has joined with a second amino acid connected via a peptide bond. We now have a polymer. And produced, of course, we already know from before, is going to be water. All right. Now, proteins, we know how they react. We know what a monomer is like. We know what a polymer is like. Proteins can have 4 levels of structure. The first level of structure is called a primary structure. And this is just the order of the amino acids that are linked together. It's very simple, right? The different types of amino acids, you start with one type of amino acid, maybe that's arginine, that is going to react with the second type of amino acid, maybe that's valine, that is going to react with the third type of amino acid, maybe that's isoleucine, maybe it's another isoleucine, maybe it's valine again, maybe it's a new one after that. The order of amino acids is the primary structure. So very simple, just the correct order of amino acids. The secondary structure is going to be called helices and pleated sheets. So a helix is a spiral, and a pleated sheet is like folding of a fan. So these different amino acids are actually going to fold up or wind up in a very interesting way to form these pleated sheets or to form these helices. The tertiary structure is going to be the complex foldings of the protein chain held together by bonds. So imagine that this entire primary structure is just like a string. And what's going to happen is the string, imagine like taking a wire and just mashing it together, you're going to get a big tangled mess. That is going to be what the tertiary structure looks like. And then we have the quaternary structure when multiple protein chains link themselves together. Okay, so the primary structure, the order of amino acids, you see that here. Secondary structure might be a helix or a pleated sheet. The secondary structure is going to be a tangled mess of just folding. And then if we have multiple of these chains put together, that is the quaternary structure. So as you can see, proteins have a very interesting shape. It looks sort of like a tangled mess, but it's actually a very purposeful shape. And that shape is really important because that shape allows the protein to do its job. Okay, what are the 4 layers of folding for a protein? Explain each one in your own words. So do all 4 primary, secondary, tertiary, and quaternary. Okay, and finally, the last thing we're going to go over today is the shape of proteins. The shape of proteins is very specific. However, in certain conditions, that 3-dimensional shape that proteins have, it can fall apart, and that is called denaturing. You may want to write this down. Denaturing is when the shape of a protein comes apart. And what can cause a protein to denature? It might be heat, it might be pH, it might be salinity, it might be chemicals, it might be some other things, maybe sunlight, but these are all things that can cause a protein to denature. All right, well, that is where we're going to stop for today. If you have any questions, please let me know.